Hey everybody, this video is going to cover what I think is one of the most interesting and amazing parts of biology. And I hope that you'll leave this video with a better understanding for how genes are switched on and off. In other words, how we regulate the expression of our genes. When genes are expressed, that means the DNA in a chromosome is going to be transcribed by RNA polymerase. A messenger RNA will be made, and then the product of that gene, typically a protein, gets made out on a ribosome. So the gene is switched on, and the cell makes a protein that it wasn't making before, and that's gene expression. And why is this important? Well, it is absolutely mind-boggling to me that, for example, I have muscle cells in my body that have all the instructions for how to be a brain cell. And likewise, those brain cells have all of the instructions in their chromosomes for how to be a human liver cell. So why are my muscle cells acting like muscle cells and not brain cells? And why don't they act like liver cells? Because they both have the genes that are the necessary instructions for how to be a liver cell too. This is true for all multicellular organisms. I'm looking out at the trees right now, and the few leaves that are left on the trees, those leaves have the genes in their nuclei for how to be a cell in the stem of the plant or in the root of the plant. So this is true for all multicellular organisms. And so on a moment-to-moment basis, these cells have the instructions for how to be any type of cell in the body of the organism. And in terms of development, we all start as a fertilized egg, a single, undifferentiated, sort of round blob of a cell. And that cell has all the same instructions that are in the nuclei of all the specialized cells that come after. So how does this happen? How is it possible that we can have cells in our body that all have the same nucleus, but those cells behave very, very differently? Well, it depends on which genes are being expressed, which genes are switched on and switched off. In a liver cell, the liver cell genes are switched on, and the genes for controlling how a brain cell acts are switched off. So how does that happen? Like lots of things in biology, we have learned about biological processes by first studying the simplest organisms, like the bacterium E. coli, Escherichia coli. And E. coli is all over your body right now, in your mouth, in your intestines, all over your skin. It is with us all the time, billions of them. And E. coli is one of the bacteria that we have studied the most in the past, and so we understand its processes and systems quite well. And by looking at E. coli, we get insights into how all organisms control gene expression. And it doesn't work exactly the same, but the models that we'll look at in bacteria give us a really good indication for how things work in more complex organisms like us. So we're going to look at two different systems in E. coli, the TRP or Trip operon and the LAC or Lac Operon. An operon in bacteria is a group of genes that sort of function as a collective. They operate together to perform specific functions in a cell. And we'll look at two of those operons and see how those genes that are in the operon are controlled. So a busy diagram here, but stick with me and hopefully we'll figure this out. Two sections, the top section and the bottom section we'll talk about separately, but first a little bit about the Trip operon. The Trip operon is responsible for making the amino acid tryptophan. So 20 different amino acids that we need to build our proteins. E. coli, more or less, uses the exact same amino acids. And E. coli has this collection of genes in the trip operon that make the enzymes and proteins necessary to synthesize tryptophan. So if the bacterium needs tryptophan, it can make some. If it's got all the tryptophan that it needs, well, then the cell would not want to switch on these genes. It would not want to transcribe the messenger RNAs and then translate those mRNAs into these proteins. Why bother making all these proteins if you don't need the products that those proteins make? So, not surprisingly, the switch for turning on and turning off the trip operon is the tryptophan molecule itself. In the top, we're looking at the situation where the cell does not have tryptophan. In that case, we have this repressor protein that the cell has in the cytoplasm all the time in an inactive form. When the repressor protein is not active, it cannot bind to the DNA, and if that repressor is not bound to the operator region up here on the DNA, then the enzyme RNA polymerase can slide right on by, transcribe those genes, and the cell can make those proteins. So at all times in the cell, this protein is hanging around, and if there's no tryptophan in the cell, the protein stays in an inactive form, does not bind to the DNA, and the enzymes in the trip operon can be produced. So when would the cell switch off those genes? When would it stop making the proteins needed for synthesizing tryptophan? Well, if there was lots of tryptophan in the cell, it would want to switch off these genes so that it doesn't make extra of these proteins. And it's tryptophan itself that is the switch. If there's lots of tryptophan in the cell, the repressor protein that we talked about in the first part up here, that repressor protein has a binding site for tryptophan. So if there is tryptophan in the cell, some of it will lock on to the repressor protein. When that happens, it changes the shape of the repressor, and now this active repressor protein becomes what we generally call a transcription factor. A transcription factor is something that binds to DNA and either activates or inhibits messenger RNA production. It inhibits the transcription of genes. So if there's plenty of tryptophan in the cell, the protein that now has tryptophan attached to it can bind to DNA, and it physically blocks the RNA polymerase enzyme from transcribing the genes. So the genes are switched off. So no tryptophan in the cell, let's make those proteins to make tryptophan. More than enough tryptophan in the cell, those genes for making tryptophan get switched off when this repressor protein binds and blocks transcription. So transcription factor is a general term used for any molecule that binds to DNA and either activates or represses or inhibits transcription. The Lac Operon is controlled in a similar way, only this time the interaction between the repressor protein and the product that we're dealing with, the molecule that we're dealing with, is a little different. So the Lac Operon, you can see fully here on the bottom part of this diagram, is made of 3 separate genes that make 3 different proteins that are responsible for being able to digest and utilize the sugar in milk called lactose. You've probably heard of lactose intolerance. Lactose is a sugar found in mammalian milk, including human milk. And so lactose is a substance that is potentially in the environment of E. coli, and it is a sugar. It can be used as energy. And so if lactose is in the environment and the E. coli needs some energy, it would want to make these proteins so that it can break down lactose and use the energy. If lactose is absent, if an E. coli is living on the surface of your skin, for example, it's not going to get any lactose. So would it want to make these three proteins that would never have a function in the cell? No, it wouldn't. And so when there is no lactose in the cell, the repressor protein that exists all the time inside the cell is in a conformation that will allow it to bind to this operator region, the same kind of thing that we saw with the trip operon. The operator is between the promoter, where the RNA polymerase enzyme can bind, and the actual genes themselves. And so when there's no lactose in the cell, repressor protein binds to the operator, RNA polymerase is blocked, and no genes are transcribed. And that's what the cell would want. Don't switch on the genes if those proteins have nothing to do. So when would these genes be useful? Well, if there's lactose present in the cell. And when that happens, like it could be a bacterium in your gut, and you just ate a nice tub of yogurt, and you've got a bunch of lactose in there, now those bacteria might want to switch on those genes and use that lactose as a source of energy. Well, if lactose gets into a bacterial cell, some of those lactose molecules would spontaneously be converted into a very similar molecule called allolactose. And if allolactose is there, that molecule binds to the repressor protein, the same one that we talked about up in the first part of the diagram. When the allolactose binds to the repressor protein, it changes shape, and it can no longer stick to the DNA. It can no longer block the operator. So if we were in this situation above and allolactose entered the cell, the allolactose would bind to the repressor protein and knock it off the DNA. With no repressor in this part of the chromosome, now the RNA polymerase enzyme can get past, and it can transcribe the genes. Those genes can be translated into these proteins, and now the bacterium can use lactose as a source of energy. So the Lac Operon and the trip operon provide examples for how these cells automatically switch on and switch off genes depending on the conditions in their environment. It's like easy to imagine that there must be some sort of controller inside the cell, right? Some little man pulling levers and pushing buttons and switching switches on and off. But it all happens spontaneously based on the conditions in the environment, inside the cell and outside the cell. And I think we should pause now because this video is long enough.